Oh, hi everyone. I'm Hazel, and welcome to this week SCSC Club. Today, I'm excited to introduce you to the next version of Talk2Data, a powerful tool designed to enhance your biological research, helping you in investigating more complex biological questions. Joining me here today is Julie, a biologist from our Talk2Data development team. So, as biologists here in bioturing, we have firsthand the challenges researchers face in analyzing and interpreting really complex biological data. And that's why we've been working like really hard to enhance Talk2Data, providing you with a more comprehensive and efficient platform. Okay. Julie, be sharing, insights into the technical advancement and also some new features that we've incorporated into this new version. So before we get started, Julie will now provide a recap of our previous session. Okay, Julie? Sure.
Okay, so from last week's talk, we investigated stromal cells in triple-negative breast cancer, and we performed more of an exploratory analysis across different platforms on bioturing to see how we can perform different types of analyses. So first, we used bbrowser X to investigate the functional pathway differences between, sorry, I see the chat box going off a lot. Okay, perfect. Okay, so we use bbrowser X first to investigate the functional pathway differences between stromal subtypes. And we found that these stromal subtypes not only express genes that may reflect the phenotypic differences, but these differences were also reflected in the different functional pathways as well. And so given that, while single-cell RNA-seq data can bring us a lot of insight into the functions of stromal cells in triple-negative breast cancer, we can further validate some of our findings by bringing in spatial data. And so this is where we analyzed cancerous breast tissue in bioturing lens. And so from spatial data, we were able to identify these same stromal subtypes and also viewed that they were spatially distinct from one another. One particular subtype, which we found to have pathways associated with angiogenesis, localized to the blood vessels in our tissues, which leads us to believe that this subtype may play an important role in tumor growth via blood vessel function. And so as a result, our findings suggest that differentially targeting these stromal cell subtypes opens up an avenue for more specific therapies related to triple-negative breast cancer. Okay, and so that's the summary from last talk. And with that said, this is only one study that we've investigated and explored. And while we may have found some interesting insights from this one analysis of a single public study, the biggest challenge remains, which is, reading up on more studies and seeing if our findings still uphold. And I think this is a good segue to talk about the next version of Talk2Data, which utilizes this idea of bringing together multiple studies to perform an analysis so that we can more confidently construct a narrative that reflects these interesting biological findings.
Okay, thank you, Julie, for your very comprehensive overview. And I also understand your difficulties while exploring different studies. Okay, so let's dive into the agenda for today's session, shall we? Yeah, okay, this is the agenda. So first, we begin by exploring the capabilities of Talk2Data, like the current version of Talk2Data, and also its potential applications, and then we'll dive into the beta version of the platform using some research questions related to inflammatory bowel disease or IBD as our main focus. Specifically, we'll examine three questions. First is like one comparative analysis of TFF3 or the trefoil factor 3 in IBD, and then we will investigate the TFF3 and also ABOB expression in Crohn's disease. And last questions will be the Crohn's disease epithelial cells expression profiling. And finally, we'll discuss our future plans for Talk2Data and how it can continue to evolve to meet the needs of researchers. Okay, so let me first introduce you to the current of Talk2Data. Okay, As you can see, let me go here. Okay, so as you can see, this is the home page of TALK2DATA. And TALK2DATA is a public database. Okay, so you can see multiple database, I mean, multi-omics data ranging from single-cell RNA-seq data, spatial, both for bulk and also for single-cell spatial transcriptomics. We also have the spatial proteomics as well. On TALK2DATA, you can perform cross-study analysis using different databases, okay? And as you can see, on like in the middle of the screen right here, we have three main functionalities of TALK2DATA, where you can, let's say this is a big library, and you can search by gene name, you can search by cell type name, and you can also search by study name. Okay, to showcase you guys to the current TALK2DATA, just in case that many of you might not be really familiar with the platform, I will perform like a little bit of introduction, like of the demo thing into the current TALK2DATA. Okay, okay. So as you can see here, let me just zoom it in so that it's easier to see. Okay, like the gene searching by gene. For example, I have like one gene of interest as I just present in the agenda, which is the trefoil factor 3. Okay, let me just type down here and we search for it. Okay, as you can see that what TALK2DATA can do is show you the TFF3 expression across our whole database, like different expression across different cell types, showing on the ontology right here. As you click onto one cell type, you can actually see like the detailed expression of this gene in different subtypes of cell. Okay, so what question can you answer using this kind of gene search? So you can like answer, okay, what is the difference of TFF3 expression across different cell types? That's right. However, we have more complex question, like how about the expression of TFF3 in one specific cell type, but different, but between different conditions, like for example, between IBD and normal patients. So based on that, like, more complex questions, we try to develop the newer version of TALK2DATA so that it can cover those type of questions, which we will be exploring today. Okay, that's about the current version of TALK2DATA. And based on this, I will bring you guys to the TALK2DATA beta version and answering more complex biological questions. Okay, and we go back to the slide. Here you go. Okay, so as you can see, this is the current TALK2DATA. And now we will dive into the capabilities of TALK2DATA beta by addressing our first research questions. How does trefoil factor 3 expression vary between individuals with IBD and those without? So how can we do that? I think that Julie here will be very happy to show me how to go to TALK2DATA beta and answer the first question.
Yeah, so in order to go to the new version of TALK2DATA, we'll open up a new tab. Okay, and you'll first land on the current version of TALK2DATA, but to access the new beta version, click go now on the top right here. Okay, and then Hazel, if you can remind me of the overall research question that we are trying to answer.
Okay, so I would like to know the expression of TFF3 in epithelial cells between IBD and normal participants.
Okay, so the first thing we'll do is query for TFF3.
Okay.
And then we click search. Okay, and then immediately you can see that we are met with this view of the cell ontology tree where you have different cell types. And then within each cell type, it kind of blooms into more child nodes of cell subtypes. Okay, such as this, these.
Okay, so now right here I can see TFF3 expressed on different cell types. So Julie, can you please explain a little bit about the meaning of this ontology, like the color and also the size of the nodes?
Yeah, So if we open up this filter panel on the right, so the size of the node represents the percentage of cells within this specific cell type, this respiratory epithelial cell type that expresses TFF3, while the color of the node represents the average rank of TFF3 within these cells. So the reason why we use average rank to represent the gene expression is because since we're bringing together a lot of data from a lot of different sequencing platforms and whatnot, we want a way to overcome these batch effects. So one way is by representing the genes as a rank within a cell. And that pretty much represents like how highly that gene is expressed within that cell.
Okay, okay. So for example, I can see right away here is that like TFF3 is quite highly expressed in the intestinal epithelial cells. And as we can see, it's the very red color here compared to the epithelial cell of the thymus, which show like yellow color, like the lower rank as compared to the intestine.
Right.
Okay. Okay, I think we can move on with our research question.
Okay.
Yeah. So now that I have the TFF3, how can I, find for the disease and also the cell type I want to look at is the epithelial cell.
Epithelial cells. Okay. So let me first switch the scale to relative if we want to get a better picture. Okay. And to query for cell type, switch to the data tab here, the search function, and then we'll query for epithelial cells.
Okay.
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 There, after querying for epithelial cells, you can see that the branch is highlighted here, which means we are selecting for epithelial cells.
Okay, great. Okay, great. Now can
Okay.
we move on to the condition selection?
Condition. Okay. So once I switch over to the condition tree, you can see under applied filters, we applied this epithelial cell filter, which means that when we're looking at the expression of TFF3, we're only looking at the expression of this gene within epithelial cells for all of these diseases that we see here. Okay.
Okay.
And the diseases or the conditions that we're interested in are IBD, right? Inflammatory bowel disease. And we also want to compare this to normal as well. Okay.
Yeah, okay. Oh, I can see that we have one question from Paramo right now that are quite interesting. Okay, so the question is, in the filter option for studies, can we do a not option for conditions or cell types? For example, look for studies that are like normal and condition A, but not in condition B. So can we do that kind of filtering here?
Sure. So we can, let's say we don't want to include, what's enterocolitis, maybe? Or no, let's do a cancer. Let's say we don't want to include cancers. Or random lung cancer. Okay.
Yeah. Oh, okay. So I can see that
Yeah.
for like we can select like different conditions and to be included, but not really
Yes.
like not included into the into the filter right here.
Excluded. Okay.
Yeah, like it is excluded from the filter.
Mm.
Oh, so I think like this might be a very good and like a question, but also a very good suggestion from Perumo that where we can actually not only to filter for do, but we can also filter for do not, like do not include condition B as well. So thank you, Peramo. I think that we will transfer your request to our development team, and that we will work on it, and we will get back to you as soon as we have any new ideas or that kind of thing on this request. Thank you so much. Yeah, okay, I think we can move on with the TFFB in IBD versus normal in epithelial cell. Okay, like as now we already said like different, I can see like conditions and also the cell type. Oh, I can see that actually the cell type move from the apply field from like the search to the apply filters. What does it mean as well?
Yeah, so whenever you switch between different ontology trees, so since we're no longer in the cell type ontology tree and we switch to the condition ontology tree,
Mm.
that means we're looking at TFF3 expression in epithelial cell types only. Okay, within
Yeah.
this tree.
Okay. Okay. Then now how can I know the difference expression of TFF3 between the two conditions?
Yeah, okay. So to get a more simplified look, we can switch over to the heat map.
Okay.
Okay, and this will directly compare our conditions of interest. Let me switch these two levels so it's more simpler to understand. Okay. Right, okay. And so we see within epithelial cell types, we have two different conditions, and we see that TFF3 expression is quite high in IBD compared to normal.
Okay, okay. Let me take a look first. Okay, so I can see that TFF3 is expressed in like 61.61% of the epithelial cell in IBD, and like only express, let me see, this is small. Okay, only expressed in like 16% of epithelial cell, normal epithelial cell. So actually this align with my, I think my knowledge about TFF3 before, which like, you know, in the inflammation state, the expression of this gene will increase. Okay, let me check a look. So as I can see here, we have a lot of numbers, a lot of figures here. Can you explain a little bit about the number on this actual bubble heat map? Whether it's like relate to the number on the like the dendrogram right here?
Okay, yeah. So to break it down for you guys, when you hover over a node, all of the information pertaining to this will relate to the expression of TFF3. So if we look, we see that 61.61% of epithelial cells in inflammatory bowel disease express TFF3. Whereas when we look at the percentages on the dendrogram right here, we see 6.9 million cells within the database are epithelial. Of those 6.9 million cells, 6.8 express the normal phenotype, whereas 96.3K express the IBD phenotype. So this 1.4% represents the percentage of epithelial cells that express the IBD phenotype.
Okay, so basically the number of normal epithelial cell in our database really huge compared to the number of IBD epithelial cell. Okay, great. Okay, I can see one question coming from Nabi right here, which is so interesting. Like he wondered that this heat map right here, like including epithelial cells throughout the body. Like, don't we want to compare intestinal cells only? This question, Nabi, is very interesting because actually we will try to also include the intestinal cells only in the next part of our session. And yeah, we will come with you right away. So for the
Great
next
segue.
question, for Julie right here. Okay, now we have like two levels on the heat map, the cell types and the condition. Whether we can add more levels, like more ontologies, for example, the sampling size, like what Nabi is mentioning here, and also how many levels can we add on?
Okay, so when we're in this heat map view, you can add up to 3 levels or 3 different types of ontologies. This is because we want to keep the heat map quite simple. The heat map is just to give you guys an overview of your data and to find some quick and to query some like quick gene expression. So if you want to add another level to the heat map, we have to go back to the ontology tree and add some more ontologies to the filter over here so that we can add more levels into the heat map.
Okay, great. Okay.
Okay,
Okay.
and so before we move on to the next section of adding more ontologies, let me go back to the slides, see if we can summarize. Okay, right. So we see that, we see TFF3 expression is highly expressed.
Oh, wait a minute. Let me,
sorry, give me 1
I
second.
think there's a little bit of lagging in the between. Let me just, okay. Okay.
Okay, you guys can see the
should do
screen
now.
now. Okay, perfect. So we see higher TFF3 expression in IBD cells than we do in normal cells. Okay, and basically in this analysis, we were able to identify gene expression between two different conditions, okay? So
Mm.
that's one type of analysis that you can do on this next version of TALK2DATA.
Okay, yeah, sure. For the next question here, let me go to the next slide. Okay, actually, this question will cover NABI questions, where we will discover the differences in TFF3 and also another gene of interest, ABOB expression in small intestine, like the intestinal epithelial cell only, between the Crohn's disease patient, which is like a very famous IBD disease, and healthy controls. Okay. As like Julie just show me how we can do it in action. Now I will try to perform this question myself and see like whether I can find the answer to my question or not. Okay, let me go to here. Okay, now I have two genes. I no longer have one gene. I will try to include my gene of interest right here, ABOB. Okay, I search for two genes. Okay, and we're in the condition. I don't want to do like, IBD in general anymore. So I exclude IBD. But however, I will include the Crohn's disease. Okay. And I will also include another thing. Okay, let me go here. Enterocolitis, also inflammation in the intestine as well. And now to the question of intestinal cells only. I believe that we got the anatomy right here.
Right, this is where you can query for the tissue type.
Okay, like different anatomy part of the body. Okay, now I can search for the small intestine only. Okay, and let me check up. We got epithelial cell in small intestine between normal Crohn's disease and enterocolitis. Okay, now we go to the heat map. Okay, great. Okay. Oh, this makes sense. Now I can see that TFF3 actually, increase in Crohn's disease compared to normal. However, ABOB is in like a reverse situation where ABOB will decrease in the Crohn's disease. Okay. Oh, so Nabi, I believe that this should answer your question, where we can actually focus on, one specific tissue only, one sampling size only. Okay. Okay, so I think we can go back to the slide
Okay,
where I can, okay.
give it a second to switch.
Yes.
Might be a little laggy.
Okay, let me go. So this is like a little sum up of the question, where you can see that the TALK2DATA beta version can not only help you in investigating of gene expression between different conditions only, but can also help you to investigate gene expression between different ontologies, like different combination of ontologies. In this specific case, one cell type in like the epithelial cell in small intestine between different inflammatory diseases, and the normal. We also can have like two genes here showing a really clear expression pattern difference between normal and the disease. Okay. Okay. So of course I want to summarize again, like what TALK2DATA beta can do is to compare genes expression between different ontologies group. Okay. Now let me go a little. Let me go here, and I want to delve a little bit deeper, like in-depth analysis into the IBDs or the Crohn's disease. So what should I do next? Next, I would like to examine the gene expression profiles, not only one gene or two genes anymore. I want to actually compare the whole expression profiles of Crohn's diseases versus healthy to and and then to see what genes are upregulated in Crohn's disease and what genes are upregulated in healthy controls. So to accomplish this question, we will perform differential gene expression analysis, which is a new tool in TALK2DATA beta version. Okay.
Okay, and then I can demonstrate that for you guys.
Yeah.
All right. Give a little bit of time for the screen to load again.
Yeah.
Sorry, it's a little laggy on our part.
Yes. Thing is, it's okay. It
Okay.
works now.
Okay, so on the left-hand side right here, this little symbol represents the differential expression analysis. Okay, first, we want to give this a title, so we want to compare Crohn's against normal
Hmm.
Crohn's versus normal in epithelial cells.
Yes, that
Right.
should do.
Okay, and then we are only interested in epithelial cells in the small intestine. So we'll add that to group 1 and group 2. The cell type is epithelial, also in group 1 and group 2. Then the condition is Crohn's. Crohn's versus normal.
Yes.
Okay, and then once we've added all of our filters to this group selection right here, you can see we're met with this intersection plot. So basically this intersection plot shows us how many cells are in each group. So in group 1, we see 14,000 cells, where in group 2, we see about 190,000 cells. Right, and so these are the groups that we're comparing against.
Okay, great. So for these group, we don't have any intersection, and therefore no need of, perform the overlapping removal stuff. Okay.
Yes, that's
great.
right. So since we don't expect any intersection or any
Mm.
overlap between these two conditions because we're comparing two very different conditions.
Yeah.
But if we were to compare very similar conditions, we might expect some overlap in between these. Therefore, we can choose the overlap options that we want to keep or remove in this overlap options right here.
Okay. So I think that's good to go.
Yeah.
We can perform the analysis right now
Okay,
and see. So
run the analysis.
okay, so usually how long will it take to perform an analysis like this?
This should only take a couple seconds since we are analyzing only about 200,000 cells.
Yeah.
And it's
Like, so how about like more cells? Like, or how about million cells? How long?
millions of cells, I believe it still can perform under a minute.
Oh,
So
yeah.
still a couple seconds.
That's good to know.
Yeah.
That's very good to know. Okay.
All right, so let's investigate the results. Okay, so once we click into the results panel, we are met with a gene table, and you have a list of genes with their log fold changes and their FDR adjusted p-value. You can also filter for the table for the fold change and the p-value as well.
Okay. In here, I have one question from Nabi again. That like what expression values are you using for the differential expression analysis? Like, it is average rank expression or the log norm expression? And then how do you deal with the batch effects, because you're including cells from different studies? For this question, I believe I have a little bit knowledge, so I will try to guess that since we use we still using a very big database, so we like quite take advantage of our ranking system for this one. Is that correct?
Yeah, so one way to overcome this batch effect is by representing our gene expression via a rank inside of one cell. So we don't use the mean expression, we don't use median expression. Instead, we want to see how genes are ranked against each other within each cell in the context of other cells. So I hope that was that explained it well.
Yes, great. Okay. So that's for Nabi. I hope that Nabi this answers your questions. And if you have like any further question, please feel free to type down here. Now I think it's my turn to ask more question about this DG analysis. That besides this gene table, do we have any other plot or diagram so that I can visualize my result better?
Yeah, so if we click view full report, we can see a couple different plots. So we are met with a volcano plot first, where on the right-hand side, these orange or yellow genes are upregulated in group 1, whereas these green genes are upregulated in group 2. And then
Okay.
also you can see which specific genes in this table will be upregulated or downregulated.
Okay. I can see that next to the volcano plot, we also have a little button like the violin plot. Can you introduce me to that as well?
Yeah, so the violin plot is for if you want to look at specific genes. So for example, if we click this SIRF1 gene, okay, we are met with the probability density plot first, where it plots the distribution of the expression of SIRF1 within group 1 and group 2.
Yeah.
And then if we add another gene to it, then we can see the violin plot, which plots both of the genes in both of the groups.
Okay, I got the idea now. I think that this actually, answers my question, since I already have the gene table showing upregulated like the SIRF one, which like, downregulated, and then I can see other genes as well, so that I can perform a very comprehensive analysis based on the DG analysis right here. Okay.
Okay, and then to sum this up, okay, so basically, in this summary slide, we also queried for our 2 genes of interest earlier for APOB.
Oh, I
and
think we
for-
should wait 1 second to, okay.
Okay, yeah, so we queried for APOB and TFF3 in this volcano plot and this violin plot. And we see that TFF3 is upregulated in Crohn's, whereas APOB is upregulated in normal.
Okay, great. Okay, now, yeah, that should do for the DG analysis, where, we can easily, like, and very quickly find differentially expressed genes among like customized cell populations defined by different ontologies group. And that is for the current features of the TALK2DATA beta version, based on my knowledge. So for what about next steps? What we plan to do for the next features that you would like to bring on TALK2DATA beta version?
So this, since this beta version is still under development, we're still actively adding new features. The first one is the co-expression analysis, where you can find the gene expression between 2 genes and see how they co-express within or across data sets and across studies. So in the current TALK2DATA, you also have co-expression analysis, but we only provide one method of measuring co-expression, which is the Jacquard score, to calculate the co-expression between 2 genes. Whereas in the beta version or the newer version of TALK2DATA will implement even more methods of co-expression, such as Pearson's regression coefficient.
Okay, I got that. For anyone who might wonder about the current situation of co-expression on TALK2DATA, you can actually go to the current version of TALK2DATA. Let me try to go here. Okay. Let me find the TFF3 again, and then you can go to the co-expression feature right here. So if you type one gene into the search box, the system will automatically find you top 10 to up to top 50 genes that are like most co-expressed with the gene of interest within our database. However, this calculation is database wise, so it's not specific to any cell type or any conditions, any ontologies. So for the next step, I hope we will be able to identify the co-expression genes, but in a very customized cell population.
Right, so if you remember, in the same way that we were able to perform DEG analysis across different conditions, across different cell types, across different anatomies, we're also bringing in that same sentiment, that same type of feature to this co-expression. We also want to find co-expression across these different ontologies.
Okay, great. How about other features as well?
Yeah, so the second feature that we have coming up is also finding marker genes for a cell population. Okay, so from my understanding, the current TALK2DATA, we have a cell type search function right here that allows you to query for one certain cell type in order to find the marker genes for that cell type.
Yeah, that's right. So for the next one, we use the concept of customized subpopulation, and we can actually find the potential marker genes for that specific subpopulation, like predefined based on the conditions or the tissues, that kind of thing.
Yeah, exactly.
Okay, great. Okay, so yeah, now we come to the summary of today. Yeah, I think
I can summarize.
yeah.
Okay. So we have three top functionalities.
Wait me a minute. I think that the screen switching is not complete yet.
Okay. Sorry about this, guys. Technical difficulties.
Yeah. Okay. It
Okay.
show up.
So to summarize, we have three top functionalities that you can use right now in the next version of TALK2DATA. The first one, you can identify the gene expression pattern between different conditions. So if you have a diseased versus a non-diseased, you can query for any gene and then see the gene expression difference between those conditions. The second functionality is where you can compare genes across different ontologies. So instead of just comparing two different conditions, you can compare two different conditions within a specific tissue, within a specific cell type, within a specific type of study. For example, instead of just single cell data, we have single nucleus data as well. Okay, and then you can compare genes across those different ontologies. And then the third functionality is you can find differentially expressed genes among all of these different ontologies as well. Okay, and then of course, to recap again, we have two future features coming up, the co-expression analysis and the marker gene analysis.
Okay, great. That sounds very potential to me. And I'm really looking forward to new features, trying new features of TALK2DATA beta version. For me, I already try, play around with TALK2DATA beta quite a lot. I believe that all of you guys here today can have a chance to try out TALK2DATA beta version after this one. I believe you will have good time with it. And just in case that, you have any questions or any comments, we love comments at this point of the development phase coming from you guys. We're really happy to collect all of it. And yeah, now I think we go to the Q&A section. And we also have the little quizzes showing up right here. I hope that you can fill it out so we can prepare like better SESC club for you guys weeks after weeks. Oh yeah, please send us any questions that you may have, and also fill in these little quizzes. Thank you so much.
Also, please let us know if you enjoy this format as well, this kind of discussion format.
Yeah.
Helps us improve our talks.
Yes. Okay. Now we will wait for the questions here.
We'll stick around for the Q&A.
Yeah. And let me check. Okay, so we got one question here. Is there information on how the data was collected or where each sample came from?
On how the data was collected. Yeah, so basically our data curation team here either gets requests from other users to upload public studies. We do have a documentation page on how exactly each public study is curated, but basically we manually curate each public study. We look at it, we grab the data, we upload it, and we try to replicate the author's cell type annotation as well as their filtering as well. Yeah, so that's how we curate publicly available data.
Yeah. Okay, I think that answers the question. Okay, so we got the next question coming from Maria. Oh, first of all, thank you, Maria, so much. Maria said that this is very interesting new functionality. And she asked whether will the beta version replace or complement the functionality in the current TALK2DATA?
It will definitely complement the current features of TALK2DATA. We have no plans on replacing the current version of TALK2DATA soon because the current version still has a lot of functionalities that are very useful to our users right now, especially if you're trying to find specific studies and filter for specific studies. Basically, this beta version, this newer version of TALK2DATA will complement the old version by allowing like access to a quicker way to query for gene expression across these different types of ontologies.
Okay, great,
Yeah.
great. Okay. Okay, again, the current and the beta version will stand together and make a better TALK2DATA.
Yes.
Thank you again, Maria. Yeah. Okay. Do you have any other questions? I think we'll wait for like 1 to 2 minutes and we should finish our session today here.
Yeah.
Yeah.
Okay. Well, I guess if there are no other further questions, feel free to email us if you have any specific questions. You can also ask for a demo as well. Our team would be happy to reach out.
Yes, yes, absolutely. Please feel free to send us any questions or again, any comment about this. We hope you have a great time exploring TALK2DATA and we love receiving feedbacks from you guys. And yeah, goodbye and see you later. See you next week.
Bye, everyone.
Yeah. Okay, so goodbye, anyone. See you.